Hello everyone, welcome to our next chapter, which is talking about biological molecules. So today we'll talk about an introduction to macromolecules, carbohydrates, lipids, proteins, and nucleic acids. So macromolecules are huge molecules that are also called polymers. They consist of many similar building blocks linked by covalent bonds. Macromolecules are made from building blocks called monomers. So the entire structure is a polymer, but just one little subsection of it is called a monomer. And we have glycogen right here. Polymer right here, big section, and then one of these little squares is a monomer. So in this case, glycogen is a polymer, while glucose is a monomer. All right, cells have thousands of different types of macromolecules. Each type of cell is different in its collection of these molecules. The diversity of these molecules comes from many different options from arranging monomers. This over here is an amino acid sequence, and it then forms into a protein through folding and such like that, which we'll get into later. So polymers can be broken down by hydrolysis or water breaking. So right here, that adds a water molecule in between here, and so you get hydrolysis, this thing breaking apart. And then polymers can be built back up through dehydration, so removing that H2O right here. Okay, so macromolecules can be sorted into four main classes, which we've already gone over, but those are carbohydrates, which normally give you quick energy. Fats, which give you energy, which is very slow energy, and insulation. So these are oils and fats. Proteins give you structure, and then nucleic acids give you genetic material. And that's a picture of protein, of course. Anything that was once living you can get from nucleic acids. So going in strong with the first one is carbohydrates. Though they provide much of the energy that cells require, and they're built of simple sugar molecules. So your bread, pasta, fruits, potatoes, they are all high in carbohydrates. So it's not just a normal thing that you think of with like breads and pasta. Fruits and root vegetables have a lot of sugar in them, and so they are full of carbs. So monosaccharides are simple sugars that can be assembled into a chain of two or more simple sugars. So if you're looking for the monomer of a carbohydrate, it is a monosaccharide. So like glucose right here. A disaccharide are double sugars formed by two monosaccharides. If you put two glucose molecules together, you get maltose right there. And again, that's your H2, so you have two hydrogens and one oxygen, so there is your water right there. Oh, excuse me. And then we have polysaccharides, which are long chains of many monosaccharides, which is our glucose right here. You got lots of chains of them. And this is glycogen right here. So your polymer for carbohydrate is a polysaccharide. So we have storage polysaccharides serve as a way to store fuel. We have starch, which is in several animals, but also in plants. And then you have glycogen right here. So storage polysaccharides contain thousands of glucose molecules. Plants use a molecule called starch to store glucose, while animals use glycogen. All right, so organisms can build strong material from these structural polysaccharides. So we have cellulose right here, and that is found in plant cells. That's what a lot of their cell walls are made of, the cellulose. Also, like eating celery, that crunch that you hear on the outside edge, that is made up of cellulose. Glucose structure is slightly different in structure. Structural polysaccharides, which give these molecules more strength, but many organisms can't digest glucose. So, just like I said, celery right here. All celery is water and glucose. It really doesn't have anything else. And you can't really digest cellulose. That's why you chew it up so much. All right, chitin, and that's how this is pronounced. It's not chitin, it is chitin. Is another word for a structural polysaccharide. This is normally found in fungus and also in the exoskeleton of insects and crustaceans, so your crabs, lobster, all that kind of fun stuff. Chitin makes up their exoskeletons. This is algae right here to show you that it's very similar, that cellulose and chitin are very similar in their structures. And of course, this is what you hear whenever you step on a bug. You hear that crunching sound. That is the exoskeleton breaking. Nice. All right, so moving on from carbohydrates, we're moving into lipids, which are your fats and oils. So these are your fats, your phospholipids, which make up the cell membrane, and your steroids. They supply energy and are used to build cell parts. They make up the plasma membrane of cells and are found in adipose tissue. Foods that are high in lipids are cooking oils, meats, cheeses, nuts, all of that. So everything in this picture are very high in lipids. All right, so here we have our lipids. So you have your glycerol up here at the top with 3 fatty acid chains. This whole thing right here is called a triglyceride. So these are just large lipid molecules, like I said, that consist of glycerol and 3 fatty acid chains. So when you're looking at your monomers, lipids is the only one that has 2 monomers. So it contains both glycerol and fatty acid chains. And the number of fatty acid chains will differ depending on what kind of molecule you're talking about, but because this is a triglyceride, it has 3 fatty acid chains. So glycerol is a modified simple sugar, so that is this at the top. And the fatty acid chains are just chains of hydrogen and carbon. You can see all the way down, it's just a bunch of carbons and a bunch of hydrogens. So in humans, these are found in our skin. They also aid in cushioning around the around the organs, and they protect the body from heat loss. So we've got, like I said, the glycerol, maybe, there we go, and fatty acids. So, like I said, the monomer for a lipid, there's 2. We have glycerol and fatty acids, and then your polymer would be your something like triglycerides, your sterols, your phospholipids. There's lots of different polymers for lipids. But if you're ever asked for the monomer, there are 2, which is glycerol and fatty acid chains. All right, so you have 2 different types of fats. You've probably heard it before, but we have saturated and unsaturated fats. So you have your saturated fats over here, which are like your cheeses and your meats, and your unsaturated, which these are normally called your healthy fats, which include like fish, avocado, olive oil, nuts, things of that nature. And this just refers to how the fatty acid chains appear in lipids. Okay, so a saturated fat is a straight line. All the carbons are in a complete straight line. So they can have as many hydrogen bonds as they need to, and there's no double bonds. But unsaturated, and it doesn't always have to be towards the end, it can be on any part of this, but they have at least 1 double bond in this, and it causes a little crook in the structure. And then there's 1 less hydrogen bond, so there should be hydrogens right here. Most animal fats are saturated, so your lard, butter, and fatty meats, and are solid at room temperature because they can pack tightly together. So there is your butter. The double bond changes the structure of the fat, so unsaturated fatty acids can't pack together and are usually a liquid at room temperature, like your oils. The type of fat has healthy implications because a diet rich in saturated fats can contribute to heart disease. So right here, there is plaque building up in these arteries, and blood is not able to go through that. Saturated fats tend to stick in the arteries more than unsaturated because they're straight lines. So if they're going straight in through the artery right here, they can get stuck on the sides. These unsaturated can kind of bundle up and pass through the arteries where they don't stick onto the sides. And then you have trans fats, and those are created during food processing and are fats that have double bonds that have hydrogens on different sides of the double bond. So right here, this is what you saw just a minute ago. So this is a cis fat. So it goes right here, and then we have this double bond right here that makes it crook off, but we still have our hydrogen bonds up here. Now down here, this is a trans fat where we're no longer getting that little crook in the thing, or in our fatty acid chain, and we have hydrogens on either end of our fatty acid chain. These are not naturally occurring, and they can contribute to heart disease as well, because remember, we want these crooks in there so it can bundle up so we can get through the arteries better. With this being straight, I'm sure we still have the same number of hydrogens, but it's still straight and it can get stuck. Some countries, including the US, have banned the use of trans fats in food. So it used to be really popular, and we've gone away from that so that we can just get away from using unhealthy foods. Here is a phospholipid, and this is made up of, this is what makes up the cell membrane. So you have phospholipids. Those are the basis of the cell membrane. They're just a modified triglyceride. So we have a nitrogen-containing group up here, and then you have a phosphate, and then you have a glycerol. Just like we saw with the triglycerides, you have that glycerol up here, but it just adds a phosphate and a nitrogen group. And those are the polar heads, and then our non-polar tails, which means that they hate water, are our fatty acid chains. They only have 2 fatty acid chains. Oh, excuse me. The third chain is replaced by that phosphate group and the nitrogen group, like I talked about. So the fatty acid tails are hydrophobic, which means they fear water or they hate water. The glycerol with the phosphate and the nitrogen, we call this the polar head, and it is hydrophilic, which means it loves water. Phospholipids are the main component of the cell membrane, like I said. So you have a, which we'll talk about this in the next chapter, I believe, but you have a phospholipid bilayer. So you have one facing this way, and then you have another one with the fatty acid tails facing the other direction. Just like right here. Okay, cool. So you have your fatty acid, or not, excuse me, you have your polar heads up here at the top and at the bottom. So this would be like the outside of the cell. Here would be the inside of the cell. And here is your cell membrane. And you have your non-polar tails in the center of the cell membrane, kind of facing opposite of each other. So due to the hydrophobic nature of these non-polar tails, phospholipids will naturally form phospholipid bilayers in watery environments. So just saying that you place these phospholipids in a watery environment, they're going to try to close in on themselves so that this inside right here can be in a non-polar environment and shut itself off from the watery conditions. Phospholipid bilayers are what the plasma membrane is made up of in both plants and animals. So I know you've always learned that plants have a cell wall, but they also do have a cell membrane just buried underneath that cell wall. And there you go. Here's our phospholipids. And we'll talk about this more when we get into the cell membrane. But the cell membrane exhibits a fluid mosaic model. So all of these proteins that are moving around is because these actually do move around. The cell membrane doesn't just stay stagnant, it actually moves. So all these phospholipids can push by each other, and the proteins and receptors that are on the outside of the cell can also move around. All right, so steroids are also considered a lipid. They're flat and made of 4 interlocking rings. So you have your 4 hydrocarbon rings right here, and then you have different types of groups that stick out of here. This is a cholesterol molecule right here, which is considered a steroid. So steroids include cholesterol, which is right here, your estrogen and testosterone, adrenaline, cortisol, and insulin. All right, so moving on to proteins. Proteins function as a structural material or energy source, so hormones, cell surface receptors, antibodies, really anything that you need your body to do, proteins can make it. So they're found in meats, cheeses, eggs, and nuts. So everything you see right here. So the monomer for a protein is called an amino acid. They're attached to an amine group and an acid group. So you've got right here, this whole thing is an amino acid. We have an amine group right here, an acid group, and here is your R group in the center. But this whole thing right here is an amino acid. So proteins are really just long strands of amino acids. So you've got all these long strands right here. So amino acid sequence of a protein determines primary, secondary, and tertiary structures. So really it's just how we fold it. So the primary structure, which is this guy up here, you just have a long strand of amino acids. And that is the primary structure. So right there. The secondary structure forms a 3D model of that. So you kind of get a pleated sheet, or you can get an alpha helix. It depends on how they fold themselves up, but that is the secondary structure. Your tertiary structure is when that 3D shape then starts folding in on itself. So it starts conforming like that. That is your tertiary structure. And then once it finally sells into, we have multiple proteins together and they're all folded up together. That is your quaternary structure. Right there. We have structural proteins like collagen right here, which just provides mechanical support. They're usually fibrous. So we have collagen right here, which makes up your connective tissue. It's responsible for making sure that your bones and tendons are able to move and maintain flexible. And then we have enzymes, which we'll speak on enzymes later. But they're just catalysts for chemical reactions. So all they're going to do is speed up chemical reactions. So we don't die waiting for them. Yeah, so that's basically it. Enzymes are just going to speed up chemical reactions. Proteases they actually break down proteins. If you ever see something that ends in an ACE, A-S-E, it is like 9 times out of 10 an enzyme. So here's the structure of an enzyme. We have an active site. It is open. It is waiting for a substrate to come bind to it. Whenever that substrate binds to it, then we are able to do work. Sometimes they also need something else called a cofactor, but we're not going to really worry about that. Just know that this enzyme has an active site. This substrate comes into this active site, and then the enzyme becomes active. Boop-a-doo, boop-a-doo-a-doo. These are a lot of slides. Why is it not letting me go on? All right, finally. All right, so transport proteins move substances across cell membranes. One example of that is like hemoglobin. And hemoglobin carries oxygen throughout your body. So hemoglobin is your red blood cells, by the way, and it does carry oxygen throughout your body. And then we have contractile proteins. So right here we have myosin and actin, and those were what actually cause muscle cell contractions. So they help move your muscles around so that you're able to walk and lift things. And then we have communication proteins. So you have these these receptors right here that have chemical messengers that come in. One example is like insulin, and that helps regulate your blood sugar, your hormones in the bloodstream. They'll have these receptors as well to notify the cells. Okay. And then we have defensive proteins, so they protect against diseases. So your antibodies are considered proteins. So they work by binding to unknown substances such as bacteria, toxins, or viruses. Right here, it's showing your antibodies connecting to this virus right here, trying to destroy it.
Proteins play countless roles throughout the biological world. Some transport nutrients throughout the body. Some help chemical reactions to happen at faster rates. Others build the structures that make up living things. Despite this wide range of functions, all proteins are made out of the same 21 building blocks, called amino acids. Amino acids are made of carbon, oxygen, nitrogen, and hydrogen atoms, and some contain sulfur atoms. Selenocysteine is the only standard amino acid that contains a selenium atom.
These atoms form an amino group, a
carboxyl group, a carboxyl group, and a side chain, all attached to a central carbon atom. The side chain determines an amino acid's properties and is the only part that varies from amino acid to amino acid.
Hydrophobic amino acids have
carbon-rich side chains, which don't interact well with water. Hydrophilic, or polar, amino acids interact well with water. Charged amino acids interact with oppositely charged amino acids or with other molecules. Primary structure. The primary structure of a protein is the linear sequence of amino acids as encoded by DNA.
The amino acids in a
protein are joined by peptide
which link the amino group of one amino acid
to the carboxyl group of another. A water molecule is released each time a peptide bond is formed. This linked series of carbon, nitrogen, and oxygen atoms is the protein backbone.
Protein chains
often fold into two types of secondary structures, alpha helices and beta sheets.
An alpha helix is a right-handed coil stabilized by hydrogen bonds between the amine and carboxyl groups of nearby amino acids. Beta sheets are formed when hydrogen bonds stabilize two or more adjacent strands of amino acids. The tertiary structure of a protein
is the three-dimensional shape of the protein chain. This shape is determined by the characteristics of the amino acids making up the chain. Many proteins form globular shapes with hydrophobic side chains sheltered on the inside, away from surrounding water molecules.
Membrane-bound proteins have hydrophobic amino acids clustered together on their exteriors, so that hydrophobic side chains can interact
with the lipids in the membrane. Charged amino acids allow proteins to interact with molecules that have complementary charges.
The functions of many proteins rely upon their
three-dimensional shapes. For example, hemoglobin forms a pocket to hold heme, a small molecule with an iron atom in the center that binds to oxygen. Quaternary structure.
Two Two or more polypeptide chains can come together to form one functional molecule with several subunits.
The 4 subunits of hemoglobin cooperate so
The 4 subunits of hemoglobin cooperate so the complex can more easily pick up oxygen in the lungs and release it in the body. Different visual representations of proteins can give us visual clues about protein structure and function.
This space-filling diagram
shows all of the atoms that make up this protein. This representation, called a ribbon or cartoon diagram, shows the organization of the protein backbone and highlights alpha helices. This surface representation shows the areas in the protein that are accessible to water molecules.
Most proteins are smaller than the wavelength of light. For example,
most proteins are smaller than the wavelength of light. For example, the hemoglobin molecule is about 6.5 nanometers in size. Hemoglobin is found in high concentration in red blood cells.
A typical red blood cell contains about
280 million hemoglobin molecules. The three-dimensional shapes of proteins determine their function.
The flexible arms of antibodies
protect us from disease by recognizing and binding to pathogens and targeting them for destruction by the immune system.
The hormone insulin is a small stable protein that can easily maintain its shape while traveling through the blood
to regulate blood glucose level.
Alpha amylase is an enzyme
that begins digestion of starches in our saliva. The calcium pump is aided by magnesium and powered by ATP to move calcium ions back to the sarcoplasmic reticulum after each muscle contraction. Ferritin is a spherical protein with channels that allow iron atoms to enter and exit depending upon an organism's needs.
Ferritin forms a hollow space with iron atoms attached to the inner wall.
Forms a hollow space with iron atoms attached to the inner wall. Ferritin stores iron in a non-toxic form.
Collagen forms a strong
triple helix that is used throughout the body for structural support.
Collagen molecules can form elongated fibrils,
which aggregate to form collagen fibers. This type of collagen is abundant in skin and tendons.
Learn more about the functions and 3D
structures of
proteins and other molecular machines at the RCSB Protein Databank.
At the RCSB Protein Databank.
All right, now that we've covered all the rest of them, nucleic acids is the final one we're going to talk about. So nucleic acids make up your RNA and your DNA. So nucleic acids are the molecules that actually control cell activities and determine what protein the cell makes. There are two main types of nucleic acids. We have deoxyribonucleic acid, which is DNA for short, and your ribonucleic acid, which is RNA for short. So the monomer, or the small puzzle piece of nucleic acids, is called nucleotides. You have adenine, shortened to A, guanine, shortened to G, cytosine, which is shortened to C, thymine, which is shortened to T. Those are all for DNA. The first three are the same for RNA, but instead of thymine, you have uracil in RNA. So nucleotides are composed of a nitrogen base, a sugar, and a phosphate group. I was hoping it would show you a picture, but it didn't. But like I said, these are the 4 main types for DNA. You have adenine, guanine, cytosine, and thymine. Bases and hydrogen are bonded together by hydrogen bonds. So in between each of the bases right here, you can see the different colors, but in between each of those, they are connected by hydrogen bonds. A binds to T, and they have 2 hydrogen bonds in between them, while C binds to G, and they have 3 hydrogen bonds in between them. There's 4 different nucleotides in RNA. Like I said earlier, they're adenine, guanine, cytosine, and uracil. RNA only has 1 strand, so there aren't any other bases. With your DNA, you have the double helix or the double strands. RNA only has 1 strand, so there's no hydrogen bonding between the base pairs. So these nucleotides are attached to the sugar backbone. So the outside edge of both DNA and RNA has a sugar background. In DNA, it's called deoxyribose, while in RNA, it's called ribose. That's where they get their names from. So this is, you can see right here, the sugar. Here's your hydrogen bond. The A, C, T, and G are your bases. And you can see the little dots right here where the hydrogen bonds are at. The backbone also has phosphate. All right, so this should show you. I clicked before I went too soon, but this backbone, it has the sugar, but it also has a phosphate group on there as well. But here is a video to explain it. Maybe? No? Okay, it's not working. But here you can just see, like I said, here's DNA, and you do have 2 strands of it. You have your hydrogen bonds in between them. A pairs with T, G with C. Your backbone right here is composed of sugar and a phosphate group. RNA, it only has 1 strand. And so you have your U, G, A, and C. And this still has a ribose sugar and phosphate backbone. So the function of DNA, it stores the information to make cells, or to help cells make proteins. And we'll talk about that in a couple weeks. I think we've got a little bit of time before we truly cover protein synthesis, but we'll get into that. And it serves as the main copy of the genetic information. So DNA never actually leaves the nucleus. It can be replicated like this, and we'll talk about DNA replication at a later point. But DNA never leaves the cell. It's the RNA that leaves. Like I said, we'll talk about protein synthesis and what transcription and translation are later. But that's just to show you that we go from DNA to RNA to protein. So essentially RNA, at least in your body, there are several organisms that only have RNA, and they don't have DNA. But in humans, they really just serve as an extra copy of genetic material. Like I said, the DNA never leaves the nucleus, but the ribosomes are what make proteins. And so the DNA needs to get its genetic code out there, and it does that through help of RNA. So RNA molecules go out into the cell to be read by, not be read by proteins, be read by ribosomes in order to synthesize proteins. So studying all the DNA in an organism is called genomics. So looking at all the DNA that they have. So proteomics is the study of the structures of all proteins an organism or cell make. This is a revolutionized research just recently. Well, it's recently to me. Most of you were born at this time, but the early 2000s, we did the Human Genome Project, and that's how we know where chromosomes, or what our chromosomes look like, what our DNA is supposed to look like, and that's why a lot of you are able to do the 23andMe and the Ancestry.com to see where you're from, because of that Human Genome Project that happened in the early 2000s. So because of that, and because we've been able to do that research, we have now gone forth in our studies of evolution, conservation biology, paleontology, medicine, all that fun jazz. Next lecture, we're going to talk about the cell. So we'll talk about all the different organelles, but next week you have an exam. So you'll have the first exam for this semester. Next week will be for all of the things that we have covered so far. We've covered 4 different topics. I'll give you more information on the exam next week, but you'll have the exam, and then next week you will also learn about the organelles in the cell. But for this week, you just have the Chapter 5 reading quiz, and then there's also a molecule chart that you need to fill out that is in Blackboard. I'll see you guys next week. Email me if you have any questions. Bye.